Hello, my name is Katie Robinson and this is Zach Verdun. We're going to give you some general information about Anderson University's Center for Cancer Research and demonstrate some common procedures we use here. The Cancer Center is located at 813 Fant Street, right across from ANMed Hospital, and use of the building was granted by ANMed in 2011 and has been greatly utilized by students and professors during the past few years. The building is equipped with state-of-the-art research equipment that allows students to pursue sophisticated research projects that can contribute to their understanding of cancer biology.
Getting involved in research at the Cancer Center requires a few things. After taking an online research methods course, you may apply to be a cancer scholar by contacting Dr. Diana Ivankovic. Up to 4 student researchers and 4 volunteers are accepted into the Cancer Scholar program each semester. After a student is selected, they must complete 20 hours of volunteering at the Cancer Center. This is followed by a year-long research project in which the student completes an independent project of their own. With the completion of this project, the student will earn school credit and present their project to both faculty and students. Cancer scholars will also have the opportunity to travel to various conferences and present their work to other members of the scientific community.
Zach and I both completed our senior research projects as a part of the Cancer Scholar program. We spent 3 semesters learning and performing the various techniques and protocols typically involved with cancer research. It's definitely a group effort, and students learn to collaborate with professors and students alike to achieve their goals. This accomplishment starts with understanding the basics of tissue culture and various other techniques that are commonly used in a lab of this sort. Through a series of videos, we'll give you some important information and demonstrate techniques that will help prepare you for your time here.
First, we'll do a walkthrough of the building to orient you to the workspace and to introduce you to some of the equipment that you'll use during the research project. Because these projects require a lot of time and responsibility, you'll be provided with your own key to the Cancer Center. It is important to sign in and out every time you enter the building to work, and to always work together and never alone. We've got the -80 freezer right here. What we do is we store a lot of our cells in it because we don't want to keep them growing if we're not going to be doing stuff with them or performing experiments with them. So what we do is we take them, we follow a freezing protocol, and we put them in there, and that way they can stay there indefinitely as long as we really need. This one is kind of small. We've got it just for the stuff that we need for the Cancer Center itself. At each time, we usually have about 4 Cancer Center scholars working with us. So this is all we really need. We also have one in the biochemistry lab back at the main campus, and we do store some things in there more long term, but this is where we're going to store most of our cell cultures.
So you need to be careful about where you dispose of all of your waste, and there are a few different places that we do that. So right here in this container, you're going to open the top, and this is where we're going to dispose of anything that has been stained or dyed. So when you do a cell count, anytime that you use the trypan blue, it's always going to go in here. And this container here is for organic waste, but we're not going to put anything that has been associated with the cells directly in here because that has to be autoclaved. This is the autoclave. It's a piece of equipment that we use to sterilize things that have come in contact with the cells. So beakers, flasks, anything that you've used for your cancer cells are going to go in here to be sterilized. So just make sure that there's water in the bottom of it and that the drain is closed. And then you're going to pull this latch down to lock it. And we run it for 60 minutes. And it uses temperature and pressure to kill the cells. So after they've been in here, you pull out the biohazard bag and mark it as done, and then you can just throw it in the trash.
This is our plate reading spectrophotometer. We utilize this in order to read 96 well plates that we prepare in the tissue culture room. And what we're looking for is the absorbance reading of it. We're wanting to look at and test the cell viability through analysis of the concentration of NADH and NADPH in the cell culture media.
this is just the basic refrigerator for anything that needs to be refrigerated but not frozen, such as the media or your extracts. Nothing for human consumption goes in this refrigerator, and everything that you put in here needs to be labeled with the date, your name, and what it is. Alright, so we're going to give you a quick rundown on some of the most frequently used equipment in the tissue culture room. So first over here is the inverted microscope, and that microscope is for this room, and anytime you need to check your cells after you've done something with them, this is the microscope that you'll be using. And right here is just a beaker. Every time you're working under the hood, you'll bring a beaker under the hood with you to use as a waste beaker. And under the hood we have 3 pipettes, and then the boxes for pipette tips. And this is the 25 centimeter culture flask that you use to grow your cells. This is a filter. So anytime you prepare media or you add treatment to your media, you're going to use this filter to make sure that it's sterile. This is a 96 well plate, and we use these for our experiments. Anytime you're running a new experiment, you'll be plating your cells into one of these. And this is a multi-channel pipette. It's basically just like one of these, but it allows you to pipette a larger quantity at one time, and you use the same tips that you would use for a normal pipette. This is called a boat, and we use this when we're using the multi-channel pipette. We transfer the liquid into the boat, and it's easier to pick up the liquid with the pipette. This is an electric pipette min, and we use a serological pipette tip like this, and that allows you to pick up larger amounts of liquid. This box has the hemocytometers that you'll use to do cell counts. And this is the aspirator, and we'll use that anytime you need to get the media or any kind of liquid off your cells. And it has a glass tip that you'll insert right here, and then you can aspirate any liquid off of your flasks.
A successful research project relies heavily on aseptic technique. It is extremely important to keep contaminants and microorganisms out of your cell culture and off your materials in order to avoid contamination. Keeping a sterile environment, working slowly, and adopting sterile technique will help avoid run-ins with contamination. Every session should start the same way. First, make sure your hair is pulled up and out of the way so that it does not interfere with your experiment. Next, it's time to wash your hands. Be sure to lather your hands strong for at least 2 minutes in order to remove any foreign debris that could contaminate your cells or enter your sterile environment. When you are through, dry your hands thoroughly. Next, you're going to want to put on a clean lab coat. You want to do this so that you don't get any spills on you while you're doing your experiment. Finally, put on a new pair of gloves and make sure that your goggles are on tightly. You are now ready to start your experiment. A clean workspace is the most important component of sterile technique. A 70% ethanol spray kills microorganisms that could contaminate your cells. The culture hood should first be sprayed with an ethanol solution as well as everything that enters, including your hands. Once materials are in the hood, they should be kept organized, and all trash should be removed from the hood as soon as possible. Before beginning an experiment, place a 500 ml beaker under the hood and designate it as a waste beaker for trash that may accumulate throughout your procedure. All liquid waste will be discarded using the aspirator. The culture hood is designed to maintain a sterile environment by creating a negative airflow, so it is important that everything that enters the hood is cleaned before it is brought into the sterile environment. Reagents such as media and cell treatments should never be opened unless under the hood, and any reagents should remain closed unless being used. When they are opened, never touch the inside of the cap and place the cap upside down when it is not being used. When using a pipette to transfer liquid, a new pipette tip should always be used for every transfer, and it is important that the tip does not touch anything in the hood. All pipette tips, culture flasks, and anything else that comes into contact with the cells are to be disposed of immediately and autoclaved.
Now I'll demonstrate how to prepare growth media. It's important to understand that every cell line has its specific growth media for optimal growth. The instructions for preparation of this media can be found on the ATCC website. Each media has base components such as RPMI, and most have fetal bovine serum also. Some media also require that small amounts of antibiotic are added to them in order to help keep out contamination. Once all materials have been sprayed with 70% ethanol and placed under the hood, spray your hands so you can begin working. Open the filtering unit under the hood in order to avoid contamination. Remember to place the caps face up when you open a container of liquid. Using a different serological pipette for each liquid, transfer each component into the filtering unit. Make sure that the glass pipette tip does not touch anything inside of the hood or the inside of the containers. The amounts of each liquid are listed in the ATCC protocol. Make sure that the measurements are precise. The vacuum aspirator is then attached to the filtering unit and turned on so the liquids can be filtered through the 0.2 micron filter using suction. Once the liquid has been filtered, your growth media is then considered sterile. After you cap the container holding the media, label it in permanent marker with the date, your name, and the type of media. The container should be stored in the refrigerator and never opened unless under the hood. The base media should be returned to the refrigerator, and the fetal bovine serum should be returned to the freezer. To start a cell line for your experiments, you rarely have cells delivered or supplied to you in their growth phase and in liquid media. If ordered from a tissue culture supplier, such as the ATCC, the cells will come in a cryovial that has been stored in liquid nitrogen. The objective of this protocol is to take a cell line that has been previously cryo-stored and bring cells back to their growth phase. For this procedure, the only necessary reagent will be your cell line's complete growth media. Start by bringing the media to 37 degrees Celsius using a water bath. Remove the cryovial from the -80-degree freezer and place it in the water bath as well. It is imperative to keep a watchful eye on the vial from this point forward. As soon as the last ice crystal has dissociated from the cryo media, bring the vial of cells under the hood to seed into a new flask. 1 milliliter of the cell suspension should be transferred to each of the 2 new flasks, and 4 milliliters of complete growth media should be added to bring the volume up to 5 milliliters total in both of the flasks. Each flask should then be labeled with your name, the date, and the cell line. The flasks of new cells should be placed in the incubator and allowed to acclimate for at least 48 hours before any potentially membrane disrupting activities are performed on them. For those of you with adherent cell lines, you now know that your cells will be stuck to the bottom of the flask that you're culturing them in. There are a few situations that will require you to transfer your cells, like when you are splitting them or when you are plating them. As with all procedures under the hood, make sure that you sterilize all of your materials before they enter the hood, and as always, spray your hands with ethanol. Start the procedure by aspirating all of the media off of your cells. Using a serological pipette, add 1 milliliter of phosphate buffered saline, or PBS, to your culture flask. This solution will wash your cells and remove all of the remaining media from the flask so that nothing interferes with the trypsin. You will then aspirate the PBS off of your cells. Now you're ready to add trypsin to the flask. The trypsin is stored in the freezer, so you will need to bring it to room temperature. Using a serological pipette, add 1 milliliter of trypsin to the flask and gently move the flask in figures of 8 along the flat surface of the hood. Place the flask in the incubator for 5 minutes before checking the cells under the microscope. The trypsin has proteolytic enzymes that cleave the proteins responsible for the cells sticking to the bottom of the flask. If the cells are still attached to the bottom, return the flask to the incubator. The cells should not be incubated for more than 10 minutes. You will be able to tell that the cells have detached by performing a microscopy. You will notice that the cells become more round in shape and float in the media. Next, sterilize the flask and return it to the hood. Add 5 milliliters of your culture media using a serological pipette to neutralize the trypsin. The cells can then be split into new culture flasks or placed into 96 well plates.
What I'm about to show you guys is how to perform a cell density count. You'll want to do this when you're trying to determine the cell density within your tissue culture flask. For this procedure, you're going to want to have the biohazard waste disposal container, and this is where you put your empty tubes and flasks once you finish the procedure. You want to have trypan blue, and what trypan blue does is it stains the live cells, and so the dead cells will actually stain dark. The live cells will stain a lighter blue. You're going to want to have 2 Eppendorf tubes, one that you acquired the cell suspension from underneath the hood, and the other one that you will mix the trypan blue with the cell suspension in. You also want pipette tips and a pipette, and also the hemocytometer dish. So for this procedure, you'll take the 20 microliters of your cell suspension that you acquired from your original flask, and you're going to add it to a new flask that has nothing in it. So then you'll want to dispose of that pipette tip. Second, you'll want to add 20 microliters, so the exact same volume, of your trypan blue to the new flask that you added your cell suspension to. You want to get this mixed pretty well, so you're going to want to pipette up and down just to kind of mix it around. You don't want to lyse any of the cells, so as long as you pipette very gently, the cells will be fine. Next, you'll add 8 microliters of this suspension to one of the flasks, or to one of the hemocytometer dishes. So you want to gently insert underneath to where the adhesion forces will kind of draw it in and cover the slip completely. You don't want air bubbles, you don't want to overfill the slip, but you want to put just enough in so that way it fills the slip completely. After that, you'll want to look under the microscope and see if you see any good cells. And so you'll use a counter in order to determine how many cells are located in the largest box. And so there will be a 3x3 large box with more small boxes within those. The cells that you're counting are going to be in the largest box that you can possibly see. After you use that, you put it into our equation, and that will help you determine what the cell concentration is in your original flask, and it will give you cells per milliliter of your original solution.
In order to keep a cell line healthy and growing at an optimal rate, it is important to tend to them as often as required by the protocol for that particular cell line provided by the ATCC. Think of your cells as babies that need to be cared for attentively in order to thrive. Most cell lines will need to be fed every 48 hours. After 48 hours, remove the old media using the vacuum aspirator if your cell lines are adherent. After media removal, the cells will be attached to the bottom of the flask. Any dead cells that are floating in the media will be removed, and some living cells may be lost also, but the majority of your healthy cells will still be in the flask. 5 milliliters of fresh media should then be added to the flask. If your cell line is not adherent, you will not remove the media because the cells are suspended in it. You will just add 2 or 3 milliliters of fresh media to the flask with the conditioned media. Be sure to refer to the protocol supplied by the ATCC for specific amounts. Once the media is refreshed, check your cells under the microscope to ensure the procedure went well, and then return the flask to the incubator. Once cells have grown to a 75 to 80% confluency rate, they need to be split. If using an adherent cell line, first start by trypsinizing the cells. If the cell line being split is not adherent, then bypass the trypsinizing step. With the cell suspended in the media, take the volume based on a cell density count from the original flask and put it into a new flask. Next, add the appropriate volume of complete growth media to the flasks in order to bring them to desired cell concentration. These concentrations are given in the protocols provided by the ATCC. Once the cells have been split into separate flasks, check them under the microscope to ensure that the procedure went well. Label your flasks and return them to the incubator.
Hey, my name is Megan. I did my senior research at AU using the fluorescent microscope here at the Cancer Center. And so today I'm just going to give you a brief overview of how to use the special functions found on the fluorescent microscope here. Besides these things, it works like a normal microscope that you would find back on main campus. To begin, I'm going to demonstrate how to use the fluorescence box. This is what will give you the fluorescence on the microscope when you're trying to use any slide or any fluorescent stain. So all you have to do is cut it on, and you should see a lamp time that should come up. If this is working properly, and you can kind of see some light in the back, if you see that, then it's on and it's working, and you're ready to go as far as the fluorescence goes. A few other things here. We do have a special type of oil immersion for this microscope, and it's found over here in the little bottle. It's immersion oil type A. We also use glass cover slips on the slides from this microscope, and there's a box kept in here at all times. And then of course you have your normal lens paper to make sure you keep the microscope clean, especially if you use oil. Now I'm going to begin showing you some of the special features that are on this microscope located on the opposite side. And so first off, you need to make sure the camera is connected up here if you would like to use the computer to see your images. And so you just make sure it's on, you press this lever down, and then the wire should stay connected to the computer. To cut on this microscope, the power button is located on this side. Right now it's in the off position, and when I cut it on, I can change how bright I want it, and this little light knob changes over here, and you can kind of see how intense the light is going to be. And so this is for the white light feature. And as you can see, it now came up on the computer, and you can see the image there. If you would like to view it this way on the computer versus looking through the microscope, you do that by this knob right here. This one must always be pulled out for you to even be able to see through here or through the camera on the computer. This one is what allows you, it transfers it to the computer instead of through the lens. So you would just push this in. You now can't see on the computer screen. Instead, you would look through the eyepiece found on the microscope. If you want to use the computer, you'll probably have to adjust the image clarity. And so normally when I do it, I just will do it by looking on the computer screen. And you can see I'm focusing a little better there. So this is your main focus. This is your fine-tuning focus, just like a normal microscope. These two things are how you would move the stage forward and back and side to side. Now, as far as the filters go for using the actual fluorescence when the fluorescent bulb is on, which I'll cut on now, it's these filters right here. So if you pull this in front, this little knob right here, if you pull it out, you can then remove the filters. So now you see the screen, that is the slide that I have under there. So in this is 3 fluorescent screens that you can view things under, and depending on what stain you have, you're going to have to change what you're using to see it. So the first one is your rhodamine filter, and it's this reddish appearance. The Fit-C filter is with labeled with Roman numeral 2, and then your DAPI filter is not Roman numeral number 3. And you can see it says 3FI. These are the 3 fluorescent lenses. To put it back in, you pull the knob back out and you simply slide it in. and see it changes what you see under there. You can see it's red there, green, and so that depends on what filter you're looking under. Above it is this filter. This is used mostly for the phase contrast feature of this microscope, as well as some of these down here. I did not use phase microscope, so if you're doing fluorescence, you don't really need to know about this filter, except for that you do want it open. You want it in an open position. This changes the size that you're seeing under the microscope, and so if you want a particular pinpoint, a little particular spot on the slide, and that's all you want to see, you can use this one right here. But generally, if you want to see the whole field, you're going to make sure it's on this big one when it's slid in here. And just like that. And that's the first stop. All of these kind of come to a stop when you reach something, so it'll be the first stop is the one that you'll normally want to leave it on. Lastly, I'm going to show you how to open up the live shooting that I demonstrated earlier on the computer. So to open it, you click on EOS utility found on the desktop. And then you're going to click on Camera Settings Remote Shooting. That will pull up this little box on the side. And then you're going to click down here in the bottom at Live View Shoot. That will then open up what is being shown on the microscope. Any fine-tuning that you want to do for the picture, you're going to have to do on the microscope. So you can see how me changing on the microscope is changing it on the screen. And then if you would like to take a picture, you just need to simply click down here, pull up the little box beside it, and click the black button near the top, and that will capture your image. This is what it looks like. From that point, you can then adjust the color settings, and any other brightness, contrast, or editing you want to do can be then done on the computer itself.
Each research project at the Cancer Center involves a round of experiments in which a treatment is applied to the chosen cell line in an array of concentrations. The treatment type and concentration is decided by you and your research advisor prior to the start of the project. Whether it be a plant extract or some other source, the treatment usually starts in a solid form that is then dissolved into the appropriate amount of culture media. It is now in the form of what we call the treatment. First, start by aspirating the media off each well. This removes any of the old media that's been incubating for the last 48 hours with the cells. Next, add 100 microliters of PBS to each well, and then aspirate that off. This is a wash just to make sure that all of the media that could have been left over in the wells is all gone. Next, add 100 microliters of the treatment to the appropriate wells using your multi-channel pipette, being sure to add the correct concentrations to each row. After this procedure is through, return the 96-well plate to the incubator for an additional 48-hour incubation period. After this incubation period, you will begin your MTS assay. After the cells have been incubated with the treatment for 48 hours, an MTS assay is run to test the cell viability. The MTS assay is a light-sensitive, colorimetric assay that measures the cell viability after the cells have been exposed to the treatment. In order to run an MTS assay on the cell line, the cells need to be adhered or stuck to the bottom of the 96-well plate using some other matrix. This is to ensure that they remain in the wells during the process of the assay. First, begin by aspirating off the media containing the treatment. This will remove all of the treated media, but leaves the cells still in the wells. Next, wash each well with 100 microliters of PBS and aspirate it off. This ensures that there is no leftover treatment media still on the cells that could interfere with the MTS reading. Then, add 100 microliters of base media that the cells grow in, such as RPMI or McCoy's 5A. Be sure that it does not contain any added serum such as FBS or horse serum. The serum is a darker color than the base media and will interfere with the absorbance readings of the MTS assay. Because the MTS assay is a light-sensitive assay, Apply the MTS under low or no light conditions. Add 20 microliters of the MTS assay solution to each well and incubate for 1 to 4 hours. This is the optimal exposure time for the reaction involving the MTS assay molecule to work. The time of incubation will depend upon the cell line you use, so consult with your research advisor to determine the optimal incubation time. After incubation, read the MTS assay results using a 96 well plate spectrophotometer. Place a jump drive into the port on the side of the spec and turn it on using the switch in the back. After the machine turns on, the plate should automatically come out. Take the top off of your plate and place it in the spectrophotometer, being sure that it is placed in the correct orientation. Scroll down to the wavelength adjustment and set the wavelength at 450 nanometers, then press Accept. Press Run and wait for the spec to perform its measurements. Press the Move button and then select Export as Text. This should move the data to your jump drive. Be sure to write down the order that your data was added to the jump drive if you are running multiple plates.